YouTube.
Okay.
You can start recording.
Yes. Okay, I'll do that. Sir, I do it record on this computer or cloud?
No, cloud.
Cloud, right? Okay. Yeah. Okay, recording is also started. I think we can start, sir.
I can start. Okay. All those who are there on Zoom, I think they can respond to me, right? Hello?
Yes, sir.
Okay. Now, see, the topic is vast. It's entirely on hormones and reproduction and hormone action also. Now, either I can go through the hormones in reproduction, male and female, the hormonal control of a male and female reproduction, starting with, or I can go into hormone action. Which one would you prefer?
Hormone action, I guess.
Hormone action. So, majority agree with that?
Yes.
Yes. Okay. Fine. Okay. Should be able to get my thing. I don't want this. How do I get it? Why do I don't get my share? I do to.
It will be there down.
Yeah, share, but then it is not showing basic, advanced screen and post attendee and share sound, optimized screen sharing, etc. coming.
So you have to be a host, then only you can be sharing.
So I'm not a host at the moment. Oh, yeah.
No, sir. I have allowed sharing, so I'll again allow you. Sir, now try.
No.
Try sharing again.
Files. No. Not coming.
Because
My
only the
desktop is
host
not
can
able to
share
share.
it. Only the host can share it.
No, actually, there are settings in this, so it's possible. Just a minute, I'll check with Dinesh, sir.
All right.
Kathendra, you're from where?
I'm from Tripura.
Tripura. Oh, fine. Good. You have finished your master's or you're doing a master's?
Yeah, I finished my master. Means this is my last SAM, and I'm giving it.
Okay. All the best.
Thank you, sir.
So Dinesh sir is checking for the concern.
Okay.
And he'll enable it.
Yesterday I could share.
Darshi ma'am. Ramchandran sir,
Yeah.
the share screen is allowed, so you can share.
Okay, let me see. Let me check.
You can see the green color button and share.
Yeah, I
screen.
know, but no, but it is again giving me screen and Zoom and advanced basic, that type of a thing is opening. The Outlook meeting and all those things are opening. Not my screen.
No, but then you first open the PowerPoint presentation, sir.
Which one? PowerPoint?
Yeah, first you open that.
Oh, I see. Okay.
Then only it will show that option as a sharing.
Okay, fine. Maybe I can take it out and put it below here.
Just start the presentation,
Yeah, just a minute.
and then in the share screen you'll get that option.
Sir, can you send the presentation to me? Then I'll share maybe from
Just a
here.
minute, I'm seeing, just a minute. कहाँ गया यार? Where is it gone? Have to join again, or what is it?
No,
No,
sir.
sir.
No, but where is it gone? My screen is now seen.
So it must be minimized.
Okay, now it's come. Fine. No. Share screen, where is it now? Now share screen itself is not. Okay, share screen, they're fine. Okay, okay, what I opened will come. Okay, fine. Is it now seen?
Yes, sir.
Yeah, we got it.
Yes.
Okay,
Visible.
okay.
See? Yeah.
Okay, fine. When you talk about hormone action, I think in the morning, did any of you attend the class on signal transduction? All those who are present here for this meeting, did any one of you attend today's morning session on signal transduction?
Yes.
Yeah? Yeah, please be loud in your response. Yes?
Yes, yes.
Yeah, but then what did you go through there? So I may not repeat those things then.
So, first of all, autocrine, paracrine, all that phenomena, like how the signaling takes place. Apart from that, how the steroids can directly enter through the cell membrane and can directly take act on in the cytoplasmic receptor or the nuclear receptor. And
okay, so that was covered, all of them.
that was covered.
Okay, anyhow, I'll just go through that. What I'm saying. When you talk about hormone action, hormone mechanism and action, first thing is to understand what a hormone exactly is. What a hormone, essentially. Or how can you define hormone? What was the classical concept of a hormone?
So, hormone are small
Please be loud. You're not audible to me very clearly.
okay.
Yeah.
Hormones are small molecules, mostly protein, which can act at a site other than the source site.
In a short way, if you say hormones may not be all small molecules. Some of the protein hormones are large molecules. Glycoproteins are much larger.
Hormones
essentially, you can say they. Yeah. Who is that?
Chemical messengers.
Yeah. Essentially, refer to them as chemical messengers, maybe small or large molecules. Chemical messengers which mediate functions of organs and tissues. Right. Now, messengers, these are so-called chemical messengers, are produced by a source, that is a cell or an organ, and then you call it endocrine gland. And this hormone is released into the bloodstream. Through the bloodstream, it goes to all the tissues and organs, and the concerned target organs having the receptor for the hormone will respond and bring about some sort of biochemical change or physiological or functional effects on the target cells. So this was the classical concept. Messengers produced at one place and acting at a distant place pass through circulation. But this classical concept of hormone does not work anymore. Now we know there are certain hormones produced locally itself, we call local hormone, and in the immediate neighbor cells respond to the signal. So it need not be, so they are chemical messengers produced from a cell and acting on another cell, maybe far away or even close to its cell. So they're essentially, it's essentially for communication between cells and between parts of the body. Okay. When you talk about hormones, it is chemical or natural organic substance that's produced in minute quantity by glands to regulate growth, metabolism, and other functions of. So these are essentially what the hormones do. They can regulate growth. They can regulate metabolism. They also regulate many other functions like transport of ions and so on and so forth. So essentially, they are biochemical messengers, as we said. Now, how many types of hormones can you talk of? How many types, basically? Anyone? Hello?
Lipids and proteins.
Please speak loudly so I can hear it.
Lipids and proteins.
Lipids and proteins. Lipids and proteins. Okay. Now, can you elaborate on that? What do you mean by proteins and what do you mean by lipids? Do all lipids and proteins in general act as hormones?
No, sir.
No. Then?
Only few. For
Only
example,
few.
steroidal hormones such as
No. See, when you say a lipid, you're not answering correctly. A lipid is a macromolecule, organic macromolecule, which is defined as any substance which, when hydrolyzed, would yield fatty acids and glycerol, right?
Yes, sir.
In that concept of a lipid, every lipid is not a hormone. So lipids generally act as hormones, or they are lipid-derived hormones?
Lipid-derived.
Lipid-based hormone. All lipids are not. They are derived from lipids or lipid-related hormone. Okay. And then what about protein? All proteins are not hormones again.
Yes, sir.
Yeah.
All proteins are not hormones.
See, generally when I talk about, there are two slightly different ways of classifying hormones. If I would like to classify, I would say broadly say protein hormones and lipid hormones, fine, protein-derived lipid hormones. Under protein, you can have amino acid-derived hormones. I mean, these are hormones made of only one amino acid. A single amino acid is modified to become a hormone. That is amino acid-derived hormone. Can you give me an example of amino acid-derived hormones?
Thyroxine.
Thyroxine, okay. Then any other?
And similarly, T3. Like
T3 is again a part of thyroid hormone. T3 and T4 are the two thyroid hormones. And T3 is more potent and more active than T4. That's all. But it is essentially a thyroid hormone.
Yes.
Any other hormone which is derived from a single amino acid? Again, derived from tyrosine itself. Is there any other hormone other than thyroxine? What is the source of tyrosine in the body? Tyrosine is an amino acid. So if I ask you, what is the source of tyrosine in the body? What is the source? Tell me.
It basically gets synthesized from Krebs cycle, sir, in Krebs cycle intermediates.
Amino acid is getting synthesized from the Krebs cycle intermediate. What do you mean? Can you show me the amino acids which are being synthesized through the Krebs cycle intermediate?
Like not exactly Krebs cycle. I think PEP acts as a precursor for that.
PEP, phosphoryl pyruvate. What do they do?
Yes, sir.
Yesterday we talked about essential amino acids and non-essential amino acids. Essential amino acids are those which we need to take through diet, and non-essential those which can synthesize. So either will. So generally when you talk about the source of amino acids in the body, they are because, you take food, the food contains proteins are digested and metabolized, amino acids are released. So one of the source of amino acids is your dietary protein on hydrolysis. That's one source of tyrosine. What is the other source of tyrosine in the body? Is there any other source? Any other way tyrosine can be produced in the body? If you have studied an inherited disorder, there's one common metabolic pathway which is responsible for a number of inherited disorders. Do you know that pathway? Very common pathway that we study when we talk about inherited disorders in human beings. Inborn errors of metabolism we refer to. There's one particular pathway, amino acid metabolism pathway, which is responsible for the manifestation of number of inherited human disorders. Can you name that pathway? Metabolism of which amino acid? Ultimately, all amino acids have been metabolized, right? Extra amino acids have been broken down, completely designed into carbon dioxide. What will go ultimately? So, one of those metabolic pathways involving catabolism of the amino acid is involved in the manifestation of a number of inherited disorders. Do you know that pathway? Can we name that amino acid?
Sir, I think the amino acid will be phenylalanine.
Yes, phenylalanine metabolic pathway. That's right. Now, in that process of converting, metabolizing phenylalanine, in that pathway, number of enzyme deficiencies can result in inherited disorders. What is the first step in phenylalanine metabolism or catabolism? Phenylalanine is converted to what? The enzyme is phenylalanine hydroxylase, if you remember. Phenylalanine is converted to dash by the action of the enzyme phenylalanine hydroxylase. That means the phenylalanine is being hydroxylated. It becomes what? Another amino acid. Phenylalanine itself is an amino acid? Tyrosine.
Yes.
Phenylalanine is converted to tyrosine by the enzyme phenylalanine hydroxylase. So here is another source of tyrosine in the body. Phenylalanine itself can be converted to tyrosine. So thyroid hormones are derived from tyrosine. Any other hormone? We are not come to amino acid derived hormones. What about epinephrine and norepinephrine? Are they amino acid derived hormones?
Yes, yes.
They are produced by? Which
Tyrosine.
part of the body produces epinephrine and norepinephrine?
Adrenal gland.
Adrenal?
Adrenal gland. Cortex.
Adrenal cortex produces epinephrine, norepinephrine?
Oh, sorry, medulla part.
Medulla. Medulla is made up of what tissue? The adrenal medulla is made up of what tissue? There are some called APUT cells, APUT cells, which are nerve cells which are released during the formation of nervous system. Those cells they call APUT, they go to different parts of the body. And one part is the adrenal medulla. They're made up of nerve cells. Adrenal medulla is made up of nerve cells which produce epinephrine and norepinephrine. Any other hormone which is derived from an amino acid other than thyroid hormone, adrenal medullary hormones, anything else? Does the pineal produce any hormone? Does the pineal gland produce any hormone?
Melatonin.
Melatonin. It is derived from which amino acid? It's again an amino acid derived hormone, right? What is the starting point of melatonin? Which amino acid?
Tryptophan.
Tryptophan. So these are all examples of amino acid derived hormones. Then you have protein hormones. Now, protein hormones could be either peptide hormones or glycoprotein hormones. What is the difference between peptide hormones and protein hormones, or say glycoprotein hormone? Peptides are small, about 8 to 10 amino acids. The shortest possible peptide is a tripeptide. Any example of a tripeptide hormone that you know? That's the smallest possible peptide hormone, tripeptide. What is that? Okay, I'll come to that later. And then peptide hormones are short chains of amino acids, maybe 8, 10, or 12 maximum. Others are all long chains of amino acids called protein hormones, and many of these are glycosylated also because glycoprotein, they're large molecules. So you have amino acid derived hormones. Under protein hormones, you have either peptide hormones or protein hormones. And third is lipid derived hormones or lipoidal hormones. What are the lipid derived hormones? Or you can say
Estrogen.
steroid hormones. Steroid hormones. I'm not saying lipid. Lipid derived with different class altogether again. So amino acid derived hormones, peptides and protein hormones together in one class. And then you have steroids. Right? Steroid hormones are there. Protein hormones and steroids are a major class of hormones in the body. So what is the source of steroid hormones in the body? And which parts of the body produce steroid hormones?
So gonads and adrenal cortex.
Adrenal cortex, gonads. Okay, these are the major sources of steroid hormones. So they are referred to as sex steroid hormones and corticosteroids, right? The hormones, the steroid hormones produced by the adrenal cortex are referred to as corticosteroids, and the hormones produced by the gonads are known as sex steroids. So you have two types of steroid hormones, corticosteroids and sex steroids. Now, irrespective of the corticosteroid or a sex steroid, what is the original source of a steroid hormone?
Cholesterol.
Cholesterol. Cholesterol is the starting point of all steroid hormones. And all steroid hormones is characterized by a characteristic nucleus. It is made up of 3 benzene rings and a 5-membered ring fused with it. What do you call that? The 3-membered ring, the 3 benzene rings fused is what? Phenanthrene. And then you have a 5-membered ring fused with that. It's known as cyclopentano perhydrophenanthrene nucleus. So all steroid hormones are characterized by this central core nucleus structure referred to as the cyclopentano perhydrophenanthrene nucleus. This particular nucleus is derived from cholesterol. So cholesterol is a how many carbon atom molecule? How many carbon atoms are there in a cholesterol molecule?
27.
27. Okay. So these are steroid hormones are derived from cholesterol. Then you have lipid-derived hormone. Can you give examples of lipid-derived hormones? Prostaglandins, leukotrienes, etc. are lipid-derived hormones. Is it clear? So basically, we talk about amino acid-derived hormones, protein hormones, including peptide hormones, then steroid hormones, and lipid-derived hormones. Okay. So what we say, this presentation say they can be classified according to the chemical composition, organs where they work, example, reproductive hormones in the reproductive organs, and if they, it's okay, nothing. You think you studied about autocrine and paracrine today morning. What is paracrine? What is autocrine?
Self autocrine means in action in
Autocrines or the
Self, a
Autocrine-
cell
autocrine
responding to the
cell and
chemical
paracrine
agent it is releasing. It responds its own chemical agent. So a cell releases something, and the same cell has receptors for what it is releasing. So it acts back on the same cell itself. That is autocrine. Though the same cell is the producer of the messenger as well as the responder of the messenger, that is known as autocrine, right? Whereas paracrine is a cell is producing the chemical molecule, which is being perceived by a neighboring cell, adjoining cell. It's more like intercellular communication. That's the thing we talked about. Now, when you talk about hormones and hormone action, there are certain terms which you need to understand. What are called trophic hormones? What are trophic hormones? Please don't read and tell me. If you can just, without reading, if you can tell me. What are the very word trophic means what? When you talk about botany, study botany, you talk of lots of trophism, geotrophism, phototrophism, right? What does it mean? What does trophism mean? Trophic means what? Come on, you are master's students. Trophic means what? What is the meaning of trophic? Directed towards. A trophic hormone is a hormone directed towards something. So trophic means directed towards. Now, there'll be a word attached before trophic, and that indicates the target to which the hormone is directed. For example, I say gonadotrophic hormone. Second part, you understand very well, trophic means directed towards. The first part indicates to which organ it is directed, to the gonads. The hormone which is directed towards the gonad is known as the gonadotropic hormone, right? Similarly, you have a thyrotropic hormone, a hormone directed towards the thyroid gland, adrenocorticotropic hormone, a hormone directed towards the adrenal cortex, right? And what about growth hormone? Is there any other term that can be used for growth hormone?
Somatotropin.
Somatotrophic hormone directed towards, somato means body in general, because the growth hormone regulates the body growth as a whole. So this hormone is directed toward the body as a whole. That's what can also be referred to as somatotrophic hormone, right? So trophic hormone is a hormone that has its primary function on the regulation of another gland. Generally, when you see thyrotropic hormone, what is the source of a thyrotropic hormone?
Pituitary gland.
Pituitary gland. Is there any other term that can be used for thyrotropic hormone? Thyrotropin. In short, you can call it thyrotropin. Instead of saying thyroid stimulating, one is thyroid stimulating hormone, it's the same, or thyrotropin, directed to a thyroid, right? Now, pituitary releases the trophic hormones, and other endocrine glands in the body respond to these trophic hormones. So the major source of all trophic hormones in the body is the pituitary or the adenohypophysis. And these trophic hormones are directed towards different organs in the body, essentially glands. Now, what is the term called synergism? What is synergism? When you talk about hormone action, sometimes you have synergistic effects of hormone. What do you mean synergism? Synergism, similar. They work together to attain the same end result. That is synergism. When two or more things work together to bring about the same effect or same result, it is synergistic. They work together. Okay? So synergism is when different hormones work together and have a greater effect of all, because they're toward the same function being controlled, but two different things are trying to control. So both together, then what you call a enhanced effect or a greater effect. So when different hormones work together and have a greater effect than the individual hormone alone, is known as synergism. Is it clear? Or synergistic effects. Then you have permissiveness. What is the permissiveness? Some hormone by itself may not be acting, but its presence is required for another hormone to act. That is called permissiveness. So a small amount of one hormone allows a second hormone to have its full effect on a target cell. That is, first the hormone permits the full action of the second hormone. That means the first hormone should act. It does not bring about a major effect, but it is priming the tissue to respond to another hormone which is more potent to bring about the function. So this is called as permissive action. So many times thyroid hormones function as permissive hormone for other hormones to act. For example, in the case of reproductive system, estrogen has a priming effect before progesterone can act. When you look at the uterine development, etc., it is the estrogen which is priming the uterus. It is allowing to grow. Endometrium grows in size, proliferates, glandular systems are formed under the influence of estrogen, but it does not become functional unless progesterone acts. So progesterone brings about the functional expression of the endometrium, but it has to be primed before that by estrogen. If estrogen action is blocked, then progesterone cannot bring about the functional effects of the uterus. So they're called permissive effects also. Then you have antagonistic. One hormone may be antagonistic to another, right? They are trying to act opposite, right? So one hormone produces the opposite effect of the other. If you look into the various hormones in the body, something you heard the term all against the Rome. Have you heard the term? Something like a proverb say all against the Rome. Everybody working against Rome. So is there any such hormone where every other hormone is working against the hormone and singularly that hormone alone does its function? The classic hormone and all other hormones are working against it. Are you aware of any hormone, such hormone? It functions alone and all other hormones almost have an opposite effect. But it has to fight against all those to bring about its reflection. Very powerful hormone. A hormone related to a very common disorder.
Sir, is it insulin?
Insulin, yes. Insulin works against all. So if you look at insulin is a hypoglycemic hormone, right? It brings about hypoglycemia. It withdraws the glucose from the blood, gets it stored in the liver, muscle, and adipose tissue, right? So that's how it lowers the blood glucose level. So insulin per se is a hypoglycemic hormone, but you inject any other hormone in the body, thyroxine, growth hormone, adrenocorticoid hormone, anything you inject, everything is trying to increase. Glucagon, most important, all are trying to increase the glucose level. And so insulin fights alone against all these other hormones, which essentially have a hyperglycemic effect. That's one thing. Okay. So this is
So
called
what is an
antagonism.
example of
Yeah. Hello?
synergism?
Yeah.
Example for synergism.
Synergism, see, growth hormone, prolactin, for example. Growth hormone promotes growth. Prolactin also promotes growth. Insulin promotes growth. The number of insulin-like growth factors, some number of these factors together will have pronounced effect on the growth, etc. So that's synergism, when two function to the same effect. And then, now here we are talking about a method of classification hormone based on its solubility. Either you call it hydrophilic hormone or lipophilic hormones or hydrophobic hormones. So here, hormones have been classified into general classes based on the solubility in water. One class of hormones known as hydrophilic hormones. They're water-soluble hormones. They are transported simply dissolved in blood. Examples are catecholamines, that is epinephrine and norepinephrine, the peptide hormone, the protein hormone. They're all transported through the blood in a soluble form. That's the hydrophilic hormones. Then the second class is lipophilic hormone. They are poorly soluble in water, so they cannot be dissolved in the blood because the major constituent of blood is water. They cannot. So what happens? They must have carrier proteins to transport these hormones in the bloodstream. So they bind to plasma proteins. They got plasma protein bound. Most of steroid hormones, a lipophilic hormone, are bound to proteins in the serum. So they bind to plasma protein and it's present in the blood in a protein bound form. They're called bound form. They are lipid soluble. Examples are the lipid soluble hormones include thyroid hormone, steroid hormones, vitamin D3, etc. So this is the classification talked about. Hydrophilic hormones are proteins, peptide hormones, and catecholamines. They are primarily acting through a second messenger system, and they circulate mainly in a dissolved state in the plasma, where the lipophilic hormones are steroid hormones and thyroid hormones, and they activate genes on binding with receptors in the nucleus and largely transported in the bound form through plasma proteins. Now, this one thing which you need to understand, here he's saying primarily act through second messengers. Anyhow, this word primarily is very good here. And here they say activate genes of binding with the receptor. The original understanding about protein hormones and steroid hormones was that the protein hormones being hydrophilic, being electrically charged and larger in size, they cannot pass through the plasma membrane. So if the protein hormone has to bring about an effect in the target cell, it cannot get into the cell because the plasma membrane is a very effective barrier which would not allow any hydrophilic, electrically charged larger molecule to pass through. So a protein hormone, in order to bring about its effect in the target cell, has to bind with a receptor present on the plasma membrane, what you call a surface receptor, because the hormone cannot get inside. So by binding to a receptor, surface receptor, it conveys its message into the individual cell, and in that process, it generates what are called second messengers. The primary messenger is the hormone itself, which is not able to get inside the cell and pass on the information or bring about the effect. So it acts to a membrane-bound receptor. It binds to the receptor, conveys the message into the cell interior, and in that process, generates what you call a second messenger. It is a second messenger in turn will bring about a cascade of reactions within the cytoplasm to bring about the desired effect of the hormone, right? So in that process, now, they were said all protein hormones act through receptors. And usually they control functions in the site of physiological functions. They generally do not have a nuclear action because they do not act at the genic level. That was the original understanding about protein hormones. Even now it is true, majority of the protein hormones have their effects in the cytoplasm by activating certain enzymes, etc. They bring about physiological changes. But now we know that some of the protein hormones can also, through the cascade of signaling molecules, pass on the information message into the nucleus and also regulate gene activity, gene function. It can switch on certain genes or switch off certain genes. So the original concept of protein hormones having a non-genic action is no more true. There are certain protein hormones which also have a genic action. That means some of the messengers and phosphorylate proteins which are produced in the cytoplasm will get into the nucleus and phosphorylate some of the transcription factors and mediate gene transcription. So by and large, we can say the protein hormones have a non-nuclear effect, having effect in cytoplasm, but there are certain protein hormones, which can also bring about nuclear effect or genic effects. A similar way, we said the lipophilic hormones or the steroid hormones can easily diffuse through the plasma membrane, being lipoidal in nature. And so there is no need for a surface receptor. Once inside the cell, then they have what are called intracellular receptors. So the major difference between the two hormones, protein hormones and steroid hormones, is the protein hormones have surface receptors, steroid hormones have intracellular receptors. Now, when you say intracellular receptor, again, there are two types of intracellular receptors for steroid hormone. One set of hormones have the receptors localized in the cytoplasm. You call it cytosolic receptors or cytoplasmic receptors. And certain other hormones have a receptor directly localized in the nucleus. You refer to them as nuclear receptors. That's, I think, what you studied in the morning, if I remember, if I understand right. Hello?
Yes, sir.
Yeah.
Yes.
So tell me which steroid hormones have cytosolic receptors and which steroid hormones have nuclear receptors.
Insulin, nuclear, cytosolic.
No. Insulin is a protein hormone. I'm talking about a steroid hormone, man. We are talking about steroid hormones. And we say steroid hormones, being lipoidal in nature, can diffuse through the plasma membrane, enter into the cell. So there is no need for a surface receptor. But then they have a receptor inside the cell, either in the cytoplasm or nucleus, together referred to as intracellular receptors. For some of the steroid hormone, the receptors are localized in the cytoplasm, and for some of the steroid hormone, they are localized in the nucleus. Even for those steroid hormones which act directly in the nucleus, they first pass to the plasma membrane, diffuse through, they again diffuse to the nuclear membrane and get into the nucleus. But then ultimately, whether in the cytoplasm or in the nucleus, they have to combine with their specific receptors. Now, the question is, which steroid hormones have receptors localized in the nucleus and which steroid hormones have receptors localized in the cytoplasm? That's my question. Can you identify the hormones which have cytosolic receptors and steroid hormones which have nuclear receptors?
Glucocorticoid has cytosolic receptors, sir.
Okay, corticosteroids. Any other hormone which will have cytosolic receptor other than corticosteroid? Any sex steroid? What about androgens?
Progesterone.
What is that? Please, could you please repeat what you said?
Progesterone.
Progesterone is an androgenic hormone. What are androgens? What are androgens? Andro and gyno means what? Because the field of medical science which deals with male reproductive functions is called andrology, right? So andro is related to male. Androgens are male sex hormones. And the field of medical field which deals with the study of female reproductive system is gynecology.
Testosterone.
Testosterone is an example, okay, dihydrotestosterone, etc. They also have cytosolic receptors. What about nuclear receptors? Hormones which have nuclear receptors? Two important hormones, you tell me.
Estrogen has nuclear receptor.
Okay. Any other hormone which is not a steroid? Now, we talked about thyroxine, right? T4 and T3 are the hormones produced by thyroid gland. It's an amino acid derived hormone, right? Hello?
Yes, sir.
It's an amino acid derived hormone. Now, will thyroxine have surface receptors or intracellular receptors?
Intracellular receptor.
Yeah. So T3, and though it's an amino derived, it can pass the membrane. So it has got, and essentially, like the estrogen, they also have nuclear receptors for T3. Okay. So these are examples. Now, we said most of steroid hormone, they diffuse into the cell. and combine with the receptor in the cytoplasm like androgens or corticosteroids, and then get into the nucleus, and then this new receptor, hormone receptor complex, would bind to certain segments of DNA referred to as HRE. What does it mean? Hormone responsive element. Element means a segment, a sequence of nucleotides which can bind a hormone receptor complex. Of course, there are different HREs depending on the hormone. The androgen responsive elements, these will bind to these parts of the DNA segments will only bind to androgen receptor complex. There are corticosteroid responsive elements where this sequence will bind corticosterone receptor complex. Your estrogen responsive elements, the segments of the DNA where estrogen receptor complex will bind, and so on and so forth. So once and after entering into the nucleus, the hormone receptor complex will bind to what are called hormone responsive element, which in turn will mediate the functions of the promoter side by either increasing the rate of what do you call transcription, or reducing the rate of transcription, or sometimes even switching on a gene or switching off a gene. So at the genic level, the steroid hormones act generally. Now, this concept of steroid hormones essentially having a nuclear action or an action at the genic level is no more true. That's again exception, yeah. Like we said, generally we said protein hormones essentially have a cytoplasmic effect. Now we know, no, that's not true. There are certain hormones which can also have nuclear effect or action at the genic level. Similarly, originally we believed that steroid hormones only had nuclear effect. They only act at the genic level. But now we know even some of the steroid hormones also can act through surface receptors. This is a classic example where we always said steroid hormones have only intracellular receptors. It is not true. There are certain steroid hormones which also have surface receptors, both estrogen and progesterone. Essentially, progesterone. Have you studied about the progesterone action through a surface receptor at any time? Your master's study? No? You know one thing called oocyte maturation? Have you heard the term oocyte maturation?
Yes, sir. In
What is oocyte maturation?
Sir, maturation of immature egg that moves from what do you say, ovaries to fallopian tube.
ये क्या बोल रहे हो आप?
It's a bit
You will
confused.
corrupt my knowledge also if you say like this. Whatever I know, I'll forget. I'll get confused. You said oocyte and then ovary, fallopian tube. What is all this? We are talking about what, you tell me what is an oocyte. Okay, first let's understand what is an oocyte.
Female gamete.
Is oocyte a female gamete? Can an oocyte be used to bring about fertilization? Is oocyte fertilizable?
Only secondary oocyte is fertilizable.
Mm-hmm. And even a secondary oocyte, what is that? Okay, now let me understand the difference between a primary oocyte and a secondary oocyte. Okay. What's the difference between a primary oocyte and a secondary oocyte? I'm not able to hear. Somebody's trying to say something, but I'm not able to understand it. Yes. Now, if you ask me what, if you ask me what is an oocyte, I would simply say it's a class of female germ cell. Do you understand? If you ask me what is a oocyte, I will say it is a class of female germ cell. Is that answer correct or wrong? That means, okay, let me go on. That means there are other classes of female germ cells as well, other than oocyte. And so I'm asking you to identify or name the other classes of female germ cell. Before that, let me ask you, what is a germ cell? And where do you find germ cells? No? Students,
Cells which give rise to gamete of any organism.
okay. Germ cells are cells which will give rise to gametes, right? Yes or no?
Yes.
Are you sure or not? You said, what you said is right. That's what you said, no? Germ cells are the cells which will ultimately form gametes or which will give rise to gametes, whatever it is. Correct? That's what you want to say? Whoever
Yes,
said, please
sir.
say yes or no.
Yes, sir.
So, see, jo bola, say confidently yes.
Yes, sir.
Okay, you're right. Doesn't matter even it's wrong, but you say, you stand by it, and then you must understand why it is not correct. Anyhow, so germ cells are the cells which will differentiate to form gametes. So when you talk about cells in the body, generally we say there are two types of cells. Is it correct?
Yes.
Which are the two types of cells?
Somatic cell and germ cell.
And germ cells. All the cells in the body are somatic cells, except the cells which are present in the gonads. Where do you find germ cells? They will only find in the gonads. The germ cells are found only in the gonads because we have already said, germ cells are the cells which will become gametes. Obviously, the gametes are produced by the gonads. Obviously, the germ cells have to be in the gonads, right?
Yes.
Now, does it mean that the gonads are made up of only germ cells?
No.
No, sir.
No. There are also somatic cells, right?
Yes, sir.
Yes.
Okay, now when we talk about an ovary, a female gonad, identify the somatic cells and germ cells that you find in the female gonad or the ovary.
So the follicular cells are somatic cells.
Okay. Is there any other name for the follicular cells? More scientific name? Granulosa cells?
Yes, yes. Granulosa
Yes.
cells.
Follicle cells or granulosa cells, the cells lining the developing gamete, right? So they are the somatic cells. And within the follicle, the lining follicle cell, multi-layered follicle cells or granulosa cells, somatic cell, and inside that you have one single gametic cell, right?
Yes.
Now, when you know what is gametogenesis? You know what is gametogenesis?
Yes, sir.
What is gametogenesis?
It is the formation of the gametes. No?
So it's the process by which gametes are formed, right? You have two types of gametogenesis: the male gametogenesis, which occurs within the testes, the male gonads, and the female gametogenesis, or oogenesis, which occurs in the female gonads, right? The male gametogenesis called spermatogenesis.
Spermatogenesis and oogenesis.
Oogenesis. So the female gametogenesis called oogenesis. Now, what is the starting point of gametogenesis? Whether it is spermatogenesis or it is oogenesis. These two processes are occurring in the gonads. The spermatogenesis, the process of spermatogenesis occurring in the male gonad, the testes, and the process of oogenesis is occurring in the female gonad, that's the ovary. Now, what is the starting point of gametogenesis?
Sir, the primordial germ cells undergo continuous mitosis, and few of those cells becomes the primary oocyte or the primary spermatocyte.
Now, who is telling this? Who is telling this?
Meiosis takes.
What is that? Now tell me. Okay, let me catch. What is it? What is it?
Meiosis takes place. Meiosis takes place.
What is a young lady telling? What is she telling? बोला
Sir, meiosis should takes place.
तुम लोग है ना यहाँ कहाँ से कहाँ कूदते हो, इधर उधर सब कूद रहे हो. So naturally the concept is not clear. You're jumping from one to another. My question is very simple. What is the starting point of gametogenesis? Okay, I'll come to that. Before that, since somebody talked about PGCs, what are primordial germ cells. Now tell me, what are PGCs and where are they formed? So you said PGC, primordial germ cell, they proliferate. Where are they formed? Are they originating in the gonads? Tell me. They originate right in the embryonic condition, in the embryonic stage.
Yes. Yes.
So what is the site of origin of primordial germ cells? Where exactly in the developing embryo would the primordial germ cells be formed? Near the yolk sac area. In the developing embryo yolk sac, Yes or no?
Yes, sir.
So it is near the yolk sac area the primordial germ cells are present. They proliferate and then they migrate along the dorsal body wall to the developing gonad. So somewhere in the region of the abdominal cavity and at a particular stage of embryonic development, the gonads are developing, right? Once the gonads start developing, the primordial germ cells will migrate from the site of origin that is near to the yolk sac along the dorsal mesentery. They will migrate the chemotactic mechanism. They will migrate into the gonads and then ultimately enter into the gonad. So they are starting off from somewhere else and then populating the development. Once the gonad development starts, then only they will migrate into the gonad. Now, having said that, now can you make out which part of the egg? Now I'm talking about an egg. Egg means, say, fertilized egg or even unfertilized egg, which is fertilizable, right? Can you, looking at the structure, for example, amphibian egg, you take it. Can you pinpoint the part of the egg from where the PGCs will be formed? In all organisms, the germ cell, the part of the egg which is going to form the germ cells are already marked out. Do you understand my question?
Yes, sir.
So, in the amphibian egg, can you say there's a specialized part of cytoplasm where certain specific mRNAs, etc., present, and all those, when the egg undergoes cleavage after fertilization, that part of the cytoplasm of the egg, whichever cells come to have, whichever blastomeres come to possess that part of the cytoplasm, they will differentiate to form the germ cells of PGCs. So there's a special part in the egg itself is demarcated, the cytoplasmic mass, where there are specific regulatory molecules which will push them towards becoming germ cells. They call germ plasm. Have you heard the term germ plasm?
Yes, sir.
Germ plasm is a specialized part of the egg cytoplasm where those specific regulatory molecules are there, which will make the cells coming to possess this part of the cytoplasm into becoming PGCs. Is it clear? So the PGCs are already marked out, the part in the egg cytoplasm, because they got
So we are not able to hear you.
I think there's a connection problem. I'll just check with sir. Just a minute.
Okay, ma'am.
unmute. Okay, can you hear me now?
Yes, sir.
Yes, sir.
Okay. Where is it gone? My slide again. Okay, now you can see the slide also.
Not yet.
No, sir.
You
No,
cannot
sir.
see the slide. Again, I have to share. That's bloody problem. I have to again put it off, again share. Okay. I think I go into that. How do you put it down? Opened out. Can you see now?
No, sir.
No, sir.
क्या बात है? क्यों नहीं हो रहा अभी? Why can't you see the slide now?
So have you shared?
I shared. Share तो किया ना? Let me see. And नीचे आ जाओ. Where is my?
Yeah, it is started sharing. Yes, sir. We can see it.
Sir, unmute yourself. We can't hear you.
Sir, we can't hear you.
Sir, unmute. Sir, unmute yourself.
Trying to unmute. Okay now?
Yeah. Yes.
Yeah, we can hear.
Oh, it's a messy thing. Okay. Okay. Where we stop? I was talking about the PGCs. You know how the PGs are formed? The germplasm is marked out already in the unfertilized egg. It is that part of the settlement of the egg, which will ultimately give us the germ cell that form PGCs. The PGCs will migrate to the developing gonad. After reaching there, they proliferate, become gonocytes, and the gonocytes again differentiate to form dash cells, which will enter into gametogenesis. So you have to identify the two classes of germ cells in the two gonads, which will enter into gametogenesis. So I am asking you to identify the class of germ cell which will start off gametogenesis. So in the case of male, the class of germ cell which will enter into spermatogenesis. What is that? Gonia. Gonial cells?
Spermatogonia.
Spermatogonial. So ultimately the PGC is entered in the gonad, differentiate the early part of the embryo to become gonocytes. The gonocytes then differentiate to become spermatogonia, which will remain like that. And at the time of puberty, they are stimulated to enter into spermatogenesis under the influence of the trophic hormones FSH and LH, right? So it is the spermatogonia which enter into spermatogenesis. The similar cell, similar to that in the female gonad is oogonia, which are into oogenesis. Now, spermatogonia and oogonia will divide number of times, right? And then ultimately stop dividing and start enlarging. And they become the second class of germ cells referred to as dash and dash respectively.
Primary spermatocyte, primary oocyte.
Primary spermatocyte and primary oocyte, right? Now, it is the primary spermatocyte and the primary oocyte which will now enter into reduction division. Because, only the germ cells undergo reduction division. You have two types of cell division, mitosis and meiosis. Mitosis is carried by all the somatic cells of the body because somatic cells do not undergo meiosis. Only germ cells in the gonads undergo meiosis, that too during gametogenesis. It is during the process of gametogenesis that the germ cells undergo reduction division. So only germ cells undergo reduction division, meiosis. So the primary oocyte in the female gonad and the primary spermatocyte in the male gonad, which will enter into reduction division. Now, meiosis or reduction division is actually a combination of two divisions, right? It's two divisions combined, meiosis?
Yes.
Yes. Which is the first one? The true meiosis 1. So we are divided into meiosis 1 and meiosis 2, the two divisions. Meiosis 1 is the true reductional division where the chromosome number is halved, and meiosis 2 is a normal mitotic division. So at the end, starting from one parent cell, you get 4 because two division. First division produces 2, 2 again divides, become 4. So you should get 4 end product, 4 cells end product at the end of meiosis. But if you compare spermatogenesis and oogenesis, In the case of spermatogenesis, the primary oocyte the primary spermatocyte enters into meiosis, completes meiosis 1 and meiosis 2, and produces 4 secondary spermatocytes, right? Who is that?
Yes, sir.
Okay. So in the case of during spermatogenesis, the male gametogenesis, the primary spermatocyte enters into reduction division, completes meiosis 1 and meiosis 2, and produces 4 secondary spermatocytes. So what are secondary spermatocytes? They are products of reduction division, starting with a primary spermatocyte. Now compare the same in the case of a female gametogenesis. The primary oocyte also enters into reduction division, but it enters into meiosis 1. Now you know meiosis 1, naturally any cell division, when you talk about mitosis, meiosis, it is divided into prophase, metaphase, anaphase, and telophase, right? There are four
Yes.
phases of cell division:
Yes.
prophase,
Yes,
metaphase,
sir.
anaphase, and telophase. So meiosis also should have, only thing is a repetition. Your prophase, metaphase, anaphase, and telophase of meiosis 1, and again, almost prophase is not there in meiosis 2. It will be again metaphase, anaphase, and telophase in meiosis 2. You have to go through. Now you look at meiotic prophase and compare it with the mitotic prophase. Is there any difference between the two? The first phase of cell division is prophase. Now you compare, I'm asking you to compare the mitotic prophase with meiotic prophase. What difference can you tell? Are there
Crossing
any
over.
difference?
Crossing
Chiasmata
over.
formation,
The first difference
tetrads
that you will tell,
formation.
no.
Duration.
Yes, duration. The meiotic prophase is
Yes.
of a much longer duration compared to the mitotic prophase,
Mitotic.
right? Both of them have to pass through certain, the meiotic prophase being of a longer duration, it goes through certain substages. Can you identify the substages of meiotic prophase?
Five
One?
substages.
Yes, sir.
Which are they?
Leptotin,
Zygotin,
zygotin,
pectin,
pectin, diplotin, and dikinesis.
Yes.
dikinesis.
Okay. Leptotin, zygotin, pectin, diplotin, and dikinesis. Very good. Now, the primary oocyte enters into meiosis 1, enters into prophase 1, you call it, goes through leptotin, zygotin, pectin, diplotin, and gets arrested. It does not go to completion. So during female gametogenesis, meiosis 1 gets arrested at diplotin stage of prophase 1. This does not happen in the case of male gametogenesis. It simply completes meiosis 1, goes into meiosis 2, and produces secondary spermatocytes. That means there is something called meiotic arrest during female gametogenesis. You do not have the so-called meiotic arrest during male gametogenesis. Is it clear?
Yes, sir.
Now,
Yes, sir.
the question is why there is a meiotic arrest during female gametogenesis, why while there is no meiotic arrest during male gametogenesis? That's a question. Because anything that happens in nature, there is some purpose, there is some meaning. We are very, very often, I would not say always, and not all of us also, purposeless and meaningless. We do many things. But nature doesn't do anything wrong. When there is something, it has to have a meaning. Maybe we have not understood the meaning, right? So when we say that during female gametogenesis, there's a meiotic arrest and no meiotic arrest in male gametogenesis, there has to be a reason for that. There has to be some basis for it. Now, can you tell me what is the significance of meiotic arrest during female gametogenesis?
So because a female has to contain the entire cytoplasm into one ova.
Okay. But that is, no, wait. That difference you should talk about in terms of the end products of meiosis. We said in the male gametogenesis, we get 4 secondary spermatocytes, where at the end of meiotic division in female gametogenesis, you only get 1 secondary big oocyte. Others are 3 polar bodies. So that is with regard to the end product that is produced, unequal division. I am
Yes,
asking
sir.
about the significance of meiotic arrest.
Sir, it is because the egg has to synthesize all the factors, enzymes, which will be required in the very initial phase of the zygote formation. At that time, the cell will
Wait. Don't say in the initial phase of zygote formation. Zygote formation doesn't mean that.
fertilization.
Post-fertilization.
Yeah.
So naturally, see, now you look at this. The moment the egg gets fertilized, it becomes a very active cell. So before fertilization, the female gamete is a very idle cell. with low-key metabolic activities, just enough for survival. The moment the sperm enters into that, all of a sudden becomes a very active cell. Dynamic changes are occurring. Number of transport functions start off, DNA replication starts off, synthetic activities start off. It becomes hectic metabolic activities occurring in a fertilized egg. And immediately, along with these activities, the fertilized egg is starting to undergo division. Now, what is the type of division that the fertilized egg undergoes? It's a normal act of cell division, right? It's a normal mitotic division, right or not?
Cleavage.
It is a normal mitotic division or not?
Yes.
Yes, sir.
It is a normal mitotic division, you agree?
Yes, sir.
But why is it that we do not say that the fertilized egg undergoes repeated mitotic division? Instead of that, as I think somebody said, the fertilized egg undergoes repeated cleavage divisions. Why have you introduced the term cleavage division, though it is actually a mitotic division? It would have been simpler to say the fertilized egg undergoes repeated mitotic division. We do not say that, but we say the fertilized egg undergoes repeated cleavage divisions. Why so?
So growth of the cell does not take place.
Okay, the interface is missing.
Yes.
When does the cell grow? Between two divisions, what we call the interphase, the growth phase. Right? The two daughter cells which are just formed after division, they are smaller in size, almost half the size of original parent cell. Then there's an interphase where the cell synthesizes and grow, extra cytoplasm is produced. The cell grows in size, attains the original parent cell, again it goes into division. So there's something called an interphase between the two divisions, which is actually the growth phase, where growth activities are occurring in the cell. But in the case of cleavage division, that is gone. The interface is missing. Even before the first cleavage division is completed, the second division has started. Even before second, third, and fourth, so that the cell undergoes hectic division. That means when you say cell division, what is the major molecule that is required for cell division to play an important role? What is the major molecule that play a major role during cell division? DNA. DNA has to undergo replication. For every replication division, the DNA has to be duplicated, right? So when the cell is undergoing so fast cleavage division, no rest space at all, the poor DNA is busy replicating. It is replicating, and replicating. No rest available, right? Yes or no?
Yes.
Okay, now, having understood this much, let me ask you, when you talk about DNA, is it right if I say that DNA has two important functions? Only two important functions. Is it correct? Or do you correct me? I say that DNA has only two important functions. Would you agree with me? If you agree with me, you need to identify those two functions.
So first is it acts as a blueprint for coding genes, coding for proteins, and second it acts as a template for new
The problem is you people always brag off. Be short and sweet.
One is replication, and second is passing on the genetic information.
Transcription.
Yes, transcription.
Right? Use very simple terms in French instead of going round and round and round. So two important functions of DNA are replication, that is to duplicate, or transcription, give out the information. Right? Yes or no? These are two important functions of DNA. Yes, now we are saying immediately after post-fertilization, the egg is busy undergoing repeated cleavages. That means the DNA is replicating. Now, these two functions of DNA, replication and transcription, are mutually exclusive. They cannot occur simultaneously. If the DNA is busy replicating, it is not available to give out information. That means it is not able to undergo transcriptional activity. They are happen separately. When DNA is dividing, it is not available for transcription. When DNA is undergoing transcription, it cannot divide. It is very simple. So the two functions are mutually exclusive. They cannot occur simultaneously. Now we are saying post-fertilization, the DNA is busy dividing. It is replicating. It is not available to give out the information that the process of development, embryonic development, starting from the fertilized egg to form a two cell, the act of mere cleavage division to form a morula, what we call a solid mass of cells, and the morula forming a cavity, becoming a hollow ball of cells you call blastula, and the blastula again undergoes gastrulation with that now. Oh my God. Just a minute, please. Hello. Yeah, but I'm in that class on this GSBTM thing, net. Okay, yeah, okay. Okay, so what did I say now? So because the DNA is replicating, it cannot undergo transcription. Now, for that matter, every act of the fertilized egg, starting with simple division to form a morula, to form a blastula, to form a gastrula, you require continuous input of genetic information. In the absence of genetic information, embryonic development cannot. Starting with the first cleavage division itself, the egg cannot undergo cleavage, it cannot form a morula, it cannot form a blastula, it cannot form a gastrula. So to do all these things, you require tremendous input of genetic information to start off development. Now, the DNA has the information for these early stages of embryonic development, to how to form a morula, how to form a blastula, how to form a gastrula. But the DNA is not in a position to give out that information after fertilization. Now, how can development occur then? So development cannot occur. So what happens is the information required to carry out the initial phases of embryonic development, starting from the fertilized egg till the formation of gastrula, need to be synthesized and stored in the egg cell. They're called in the form of preformed information molecule. It is this preformed information molecule stored in the cytoplasm of the egg, which will be used for fertilization to control the early phase of embryonic development till the formation of a gastrula. So the meiotic arrest is the phase that is used during female gametogenesis to transcribe all those genes whose information will be required to control the early phase of development post-fertilization. So it is actively transcribing all those genes and also transcribing many RNA molecules and tRNA molecules, because even RNA and tRNA are produced from the gene. You have genes, right, from the DNA. So after fertilization, when number of cells are increasing, you need to increase the number of RNA molecules and tRNA molecules. There is no time for transcribing those genes from the genome after fertilization. So lots of information molecules, not only mRNA molecule, even fully formed protein molecule, rRNA molecules, tRNA molecules, ribosomal proteins, all those are being synthesized and stored in the egg cytoplasm, which can be used in the immediate phases post-fertilization till about gastrula. That means till about gastrula formation, the embryo is busy dividing, cleavage. After gastrulation, the process cell division slows down, and then the DNA becomes available for transcription. So more information will be now synthesized, which will control the process of post-gastrulation phase of development. Is it clear? So the meiotic arrest is essentially to synthesize and store the necessary information molecules and regulatory molecules required to control the early phase of development, because the DNA is not available at that time to give out the information. It is busy dividing. Have you understood the point? This is the significance of meiotic arrest during female gametogenesis, because the male gamete does not provide anything. It only provides the genetic information. It does not provide cytoplasmic mass. It does not provide any molecules for controlling development. Its duty is only to deliver the genetic material. Is it clear? So it's a very selfish cell. No, the poor female cell has to do everything. It has to store entire cytoplasm, because you require a certain minimal amount of protoplasm to form an embryo. So if the female gametogen produces again 4 secondary oocytes, the cytoplasm would have been divided into 4 cells, and the amount of cytoplasm contained in a single secondary spermatocyte will not be sufficient to form the body of an embryo. That's why during female gametogenesis, entire cytoplasm is being conserved in one cell. The secondary oocyte is a large one, 3 polar bodies are gone. Is it clear? So one thing, it is conserving the entire cytoplasm. It is also synthesizing and storing all necessary information molecules to control immediate post-fertilization phase of development. So the meiotic arrest, significance of meiotic arrest is essentially to, tell me now, is essentially to, come on. You heard so much and you cannot tell. So what is the significance of meiotic arrest? Oh my God. Hello? Am I audible to you? Okay.
Yes, sir.
Yes, sir, you are audible.
Sir,
Then why are these people not replying? I've been shouting and telling so much, and now they cannot tell anything. Now tell me, what is the significance of meiotic arrest?
I think Khushi wants to attempt this.
Sir,
attempt?
it is for the synthesis of all the factors, enzyme, and all the materials which will be required post-fertilization. because the DNA will be in the division process and it cannot.
Yeah, okay, you're right. Well, I know what you're trying to say is correct, but you're not putting exact word. That's why you have to correct and choose the correct word to express. So it become difficult. Number of words should be used less to convey the information. They essentially say the meiotic arrest is used to synthesize and store. Simply say information molecules. Simply say information molecules or regulatory molecules which are required. It could be mRNA, it could be protein, it could be anything, right? All those things are being synthesized and stored, which will control the early phases of embryonic development. Is it clear? All the information required to control the early phase of development are being synthesized and stored in the ectoplasm. That's why you have a meiotic arrest during female gametogenesis. Now, my question was something else. So the second class of germ cells are the primary oocytes, primary spermatocytes. The third class will be secondary oocytes and secondary spermatocytes. And what is the fourth class of germ cells after primary spermatocytes and secondary oocytes? What do you become?
Spermatozoa and egg.
See, the secondary spermatocytes will directly become spermatids, ootids and spermatids. The spermatids undergo a metamorphic process, undergo the dramatic transformation appearance from ordinary round-looking cell to a tadpole-looking sperm cell. So that's why you say the spermatids undergo metamorphosis. Metamorphosis is a sudden change in appearance. When you say metamorphosis, meta means different, morphosis appearance, a different appearance altogether. When the original round-looking normal cell is now looking a tadpole-like cell. Now, if you do not see this process of the spermatid getting transformed into a spermatozoon, you can never believe that this cell has come from the spermatid. Like, you look at the tadpole separately and the frog separately, and if you have not seen the process, how the tadpole gets transformed into a frog, you can never believe that this form has come from the previous form. Is it clear? So similarly here, you can never believe that spermatids are given to spermatozoa because there is a dramatic transformation appearance. This is known as spermiogenesis. So in the case of male gametogenesis, the spermatids undergo what is called a process spermiogenesis. Is nothing else than metamorphic transformation from spermatid into spermatozoa. They are functional gametes because the spermatid has to acquire all those structures and etc., which can make them motile and deliver the genetic material to the egg cell, right? Now, you do not have a comparable process in the female. The ootids simply enlarge and that's all. They store everything. That becomes the ultimate egg, unfertilized egg, what you call, right? There is no metamorphosis of ootids. The ootids are the final product which simply grow in size, enlarge, and become the egg which can be fertilized. It is only after the completion of meiosis, all right, especially in the primary spermatocyte, secondary spermatocyte, there's a process called oocyte maturation. The primary oocyte is not fertilizable. So if you take a primary oocyte and put in a culture medium and add sperms to that, the sperms will not be able to fertilize the primary oocytes. But the moment they have completed the meiotic division, they become secondary oocyte. Even the secondary oocyte, for that matter, gets arrested. You know that? The secondary oocyte has to go into the next meiosis too. It immediately goes into metaphase. There is no prophase there. Immediately goes into the second metaphase and gets arrested. Right? And that's the second arrest which occurred. That arrest will shortly, it will be terminated at the time of fertilization. As the egg is trying to undergo, it immediately finishes the second meiotic division also. Is it clear? So the oocyte maturation is essentially a process of changes which are affecting the primary oocyte so that it becomes fertilizable. Do you understand? So that is what is involved in oocyte maturation. So when the meiotic arrest is there, the oocyte is not only synthesizing and storing the information molecules required to control the early phase of development, but it is also undergoing maturational changes. The so-called maturational changes will be marked by the termination of meiotic arrest, and it goes into become secondary oocyte. So there are certain changes which occur at the genetic level and the biochemical level, or hormone level, etc., which makes these cells fertilizable. Right? The moment it completes the meiotic division, it now becomes fertilizable. And that process is called oocyte maturation. Now, this process of oocyte maturation, an important hormone is progesterone, and the derivatives of progesterone in different groups of animals. In some animals, in amphibian, progesterone directly. In mammals, etc., or fishes, etc., there are derivatives of progesterone which control oocyte maturation. Is it clear? Now, when you look at the hormonal control of oocyte maturation. They're essentially progestogen, progesterone-derived hormones, the progesterone itself. They are not acting through intracellular receptors. They act through surface receptors. Here's a classic example of a steroid hormone working through a surface receptor. That is, during oocyte maturation, the progesterone and progesterone-derived hormones in different classes of vertebrates act through plasma membrane receptors. This is an exception. So we keep our old concepts getting changed. Science keeps changing. As new information comes, you forget the old, new things you start, right? So always we said that steroid hormones do not have surface receptors. Here we come to a stage, steroid hormones also can have surface receptors. Is it clear? So here is an example where the steroid hormones have. So even in the case of certain actions of in certain cases also, there are certain surfaces. So steroid hormones, by and large, act through intracellular receptors, but they can also act through surface receptors, like what we said for protein hormone. Though by and large, protein hormones act at a cytoplasmic level. Occasionally, the protein hormones can also have nuclear action. Is it clear? That's over now? Hello?
Yes, sir.
So there is one question in the text.
What is it? Okay, now. Okay. Now look at the mode of hormone action. Generally, we say hormones work in two ways. One is it brings about the synthesis of new protein molecules, in which for the action at the genic level, they change the cell permeability. Sometimes the action of a hormone is to make the cell more permeable to certain ions, etc. The ionic channels actually open out. That's one thing. And then lipid-soluble hormones involved in mobile receptor hypothesis. What is mobile receptor hypothesis? There are two hypotheses we're talking about. One is called a mobile receptor hypothesis. The other is what we call a second messenger mechanism. The second messenger mechanism is characteristic of protein hormones because they are water-soluble, hydrophilic, electrically charged, and larger molecule. They cannot pass through plasma easily, so they use or employ second messengers inside the cell. That's why it's known as the second messenger mechanism. So second messenger mechanism is usually characteristic of protein hormone, while the mobile receptor, because here the steroid hormones enter into the cell, then bind with the receptor, then okay, now the steroid hormone diffuse to the plasma membrane, combined with the receptor in cytoplasm, and then they translocate into the mobile. They're moving continuously across the plasma membrane, through the cytoplasm, across the nuclear membrane, into the nucleus. That's why it's known as the receptor is moving from cytoplasm to nucleus. That's why it's known as mobile receptor hypothesis. Is it clear? What's the difference between the two? Say quick yes so I can go forward.
So the receptor in protein are at the cell membrane and they send secondary messenger. And in case of mobile receptor, the receptor after binding with the ligand move from cytoplasm to inside the nucleus.
Yeah. In the case of protein hormone, the receptor does not move. It remains fixed in the membrane, right? And so the major thing is they employ second messengers, which will then bring about changes. In the case of steroid hormone, they employ mobile receptors. Receptors move from one compartment of the cell to the other compartment. That's why it's called like that. It's a very general way of classifying the two types of hormone action. That's all. Okay? Just to understand the two terms. If somebody uses the term mobile receptor hypothesis, it must immediately strike you that it is with reference to steroid hormone. And somebody says second messenger mechanism, it should strike you. We are talking about protein hormone. That's all. Now look at the types of receptors when you talk about. The first part of the classification is very clear, internal, external. The protein hormones have external receptor. That means receptor localized or located on the plasma membrane. The internal receptors are characteristic of steroid hormone that is localized either in the cytoplasm or in the nucleus. When you talk about accordingly, the internal receptors could be either cytoplasmic or cytosolic receptors or nuclear receptors, and the external essentially localized in the plasma membrane. Now we look at the membrane receptor. As you said, these are the receptors which are present in the cell membrane on the surface of the plasma membrane. Now, some of these receptors are, ion channel link receptor. It is a receptor come ion channel. Basically, when the hormone binds the receptor and mediates the action, it's ion channel part. It's closely linked the ion channel. That's called ion channel linked receptor. A receptor linked with an ion channel close by, and when the hormone binds the receptor, when it gets activated, and the secondary messenger mechanism occurs inside the cell, the channel adjoining channel opens out, it permits the movement of ions. So one class of membrane receptor is ion channel linked receptor. Receptors linked with sodium channel, potassium, calcium, etc. Then G protein coupled receptor. Another type of protein receptor is a protein which is coupled with G protein, of course, on the cytosolic side or on the intracellular side. Now, when you talk about a surface receptor for a protein hormone, it's essentially a protein, right? A receptor is a protein. A receptor is a protein or not?
Yes, yes.
Yes, sir.
Now, when you say protein, does the protein have polarity? Does
Yes.
the protein have polarity?
Yes.
What do you
Yes,
mean by
sir.
it as polarity?
So the N terminal and C terminal.
Charge
The
on it.
two ends of the protein can be differentiated based on the amino group and the carboxyl group. So where the free amino group is present, that becomes the N terminal. That's suppose the anti end of the protein. The other end where you have the free carboxyl group is the C terminal, the post end of the protein. Now looking at this transmembrane. Now usually hormone receptors are transmembrane proteins. Proteins which pass through the membrane one time or many times. The largest is the 7 transmembrane domains, right? Right? So you have an extracellular domain of the protein, receptor protein, and an intracellular domain of the receptor protein. So which end or which terminus would be the extracellular domain and which terminus of the protein will be the intracellular domain? That's the question.
N terminal would be intracellular.
No. N terminus is extracellular. That is the part which is going from the ligand binding domain of the hormone binding domain. The intracellular domain is the carboxyl terminal domain, C terminal domain. Now, attached to the C terminal domain, what you call intracellular domain of the receptor, you have G proteins associated with. That is why these receptors are referred to as G protein coupled receptors or G protein associated receptors. The first one we talked about was ion channel associated receptors associated ion channels. The second is receptors associated with G proteins. Now, the question is, what is a G protein? And why is it called a G protein? Why not A, B, C, D, X, Y, Z?
Because it has affinity for GTP or GDP.
Essentially.
It
So
can
these are the
bind to- it can
proteins.
bind-
Okay. These are guanosine binding protein, guanosine nucleotide binding proteins. So certain proteins which bind guanosine nucleotides are referred to as G protein, the G standing for guanosine nucleotide, right? And what is a nucleotide? What is the simplest answer that you can give if somebody asks you what is a nucleotide? There are many ways of answering. I'm asking you to give the simplest possible answer. Remember that. The simplest possible answer for what a nucleotide is.
It has a sugar, phosphate, and
देखा
an
देखा
acid.
देखा देखा कहाँ जा रही हो? मैंने क्या बोला? तुम्हारा नाम क्या है? तुम बोला मैं यहाँ से जाता हूँ, वहां से जाता हूँ, वहां घर पर फिर नाम बोलोगे? I'm asking you, what is the simplest answer that you can give when somebody asks you what is a nucleotide?
It is a nucleoside plus phosphate.
Okay, okay. Then still doesn't tell me what it is. Okay, I ask you another question.
It is
What?
It is an inheritable molecule.
A nucleotide is an inheritable
Structural
molecule.
or
अच्छा, क्या बोल रहे हो
structural
तुम?
unit of the nucleic acid.
हाँ, क्या बोला? लास्ट में क्या बोला?
Structural unit of the nucleic
you
acid.
can say nucleotides are basic units of nucleic acid. That's the simplest possible answer you can give. I didn't ask you anything else. I asked you what is a nucleotide. You say it is a simple building block of nucleic acid, right? That's all. It's a basic unit of nucleotides. All nucleic acids are formed of number of nucleotides joined together, so it becomes the basic unit of a nucleic acid molecule. That's simple. Now I ask you, what is it made of? Then you tell me what you want to tell. Yeah. What is it made of? What is the nucleotide made of?
Nucleosides and phosphate.
Okay.
Nucleoside.
Nucleoside plus phosphate. That means it is a phosphorylated nucleoside. Right?
Yes.
Okay. And what is a nucleoside?
Sugar plus nitrogen base.
Sugar plus. What sugar is there?
Usually ribose or deoxyribose.
Ribose sugar or
A pentose sugar.
deoxyribose
Let us say
Pentose sugar.
a
Yes.
pentose sugar plus nitrogen base becomes a nucleoside. Because you have two types of nucleic acids, it's based on the difference in the pentose sugar. Ribonucleic acid is made up of ribonucleotides, and the ribonucleoside has what is the pentose sugar?
Ribose sugar.
Ribose. And the DNA has a deoxyribonucleoside, which is made of a deoxyribose sugar. So the ribo and deoxyribonucleic based on the sugar. So deoxyribose is the sugar in the nucleotides of DNA, and ribose sugar is in the nucleotides of RNA. Fine. So what position? Which is now there are three things in a nucleotide. You have three moieties or three units: sugar, nitrogen base, and phosphate. Right? Which is the central moiety in this? Which is the central molecule?
Sugar molecule.
Sugar. To the sugar, nitrogen base and phosphate are attached. To which carbon atom of the sugar is the nitrogen base attached?
The first carbon.
First carbon.
First carbon.
And to which carbon atom of the sugar is the phosphate attached?
Fifth.
Fifth. Fine. Very good. Fantastic. Fine. So G protein coupled receptors are proteins which bind guanosine nucleotides. It could be either GDP or GTP. If the receptor G protein is bound with the GDP, it is called the inactive state of the G protein. When bound with GTP, it is called the active state of the G protein. So there is a class of hormone receptors which are associated with G protein coupled receptor, or G protein. So they are called as G protein coupled receptors or G protein associated receptors. Third is enzyme linked receptors. There are certain receptors of protein hormones which are linked with or associated with enzyme on the carboxy terminus. On the intracellular domain, it is associated with an enzyme. Essentially, the enzyme is kinase associated. Essentially, it is tyrosine kinase, right? The enzyme usually that you find associated receptors are tyrosine kinases. Is it clear? Is the tyrosine kinase which can be found associated with the C terminus or the intracellular domain of a receptor. So according to what is found associated with the C terminus or intracellular domain of a receptor, you have three types of protein hormone receptors: ion channel linked receptors, G protein coupled receptors, and enzyme linked receptors, essentially tyrosine kinase. Even under that, there are two types. They're called tyrosine kinase receptors and tyrosine kinase associated receptors. You know the difference between these two? I'm saying tyrosine kinase receptors and tyrosine kinase associated receptors. What is the difference with these two terms I'm using? I use two terms now: tyrosine kinase receptor and tyrosine kinase associated receptor. What is the difference between the two that you understand?
Sir, the tyrosine kinase receptors might directly be using the tyrosine kinases, whereas the tyrosine kinase associated receptors are the receptors which work on the basis of direct and indirect association with the
I'm sure
tyrosine kinase.
you are trying your level best to confuse me completely.
No, sir.
No. See, look at, concentrate on the term that we see. Sometimes you get meaning, you just hear that sentence. Look at that consonant sentence now. Tyrosine kinase receptors. That means the receptor itself is functioning as a tyrosine kinase.
Yes.
That the C terminus domain, or the intracellular domain of the receptor itself is functioning as a tyrosine kinase. Is it clear? Whereas in the other case, the receptor is separate. Adjoining
And
that,
the
you have a tyrosine kinase. That is associated.
Yes, sir.
That's the two difference. So some of the hormone receptors are tyrosine kinase receptors, and some of the hormone receptors are tyrosine kinase associated receptors. You understand the difference between the two, right? Yes,
sir.
Yes.
Oh, yes, sir. Not forgettable, no?
No, now not.
You just go out of this class and you forget it conveniently.
No.
No, okay. So what are these hormones which use such type of receptors? The protein hormones, peptide hormones, and catecholamine. These are three classes of hormones which make use of these types of receptors. Cellular receptors, membrane receptors, which could be either ion channel linked receptor or a G protein coupled receptor or an enzyme linked receptor or enzyme receptor itself. Okay, now, what is the time? Okay, name of time maybe. Okay. Now, look at the cytoplasmic receptor, which are essentially for steroid hormone. Essentially, when you say cytoplasmic receptor, that are receptors present in the cytoplasm. Example, steroid hormones. We have nuclear receptors. We said receptors present in the nucleus, and there is a direct association with one or more chromosomes. Now, what does the sentence mean? What is this sentence trying to say? Receptors present in nucleus. That I understand. Okay. There is direct association with one or more chromosomes. What does that mean? Can you explain that? Anyone can tell me what is this? What does it mean? Okay, forget it. Now, we are here talking about the steroid hormone receptors which are localized in the nucleus, right? Now, the question is, can the receptor bind to the DNA directly? Yes?
Yes. Some
Can it bind
receptor.
directly? Receptor alone?
No, along with the ligand.
No,
Ligand plus receptor.
So naturally, the receptor is there in the nucleus, but when the ligand in the form of the cognate steroid hormone gets into the nucleus, it binds with the receptor. It becomes the hormone receptor complex. See, generally the receptor is kept inactive. See, when you say receptor, this nuclear receptor, what is the main function? What is the main function of the receptor? Of course, its functional ability is manifested only when the concerned ligand in the form of a steroid hormone binds to it. So there is a steroid hormone binding site on the receptor. When the binding occurs, that is the steroid hormone with the receptor, there's a conformational change in the steroid hormone receptor. The steroid hormone receptor is also a protein. Like the surface receptor of a protein hormone, even the intracellular receptor steroid hormone is also protein. All receptors are protein. Is it clear? So whenever something links to a protein and binds to a protein, the first thing that happens is the conformational change, the tertiary structure getting altered. Is it clear? So when there's a conformational change in the receptor because of binding of the hormone, it exposes the DNA binding domain of the receptor. So it is this hormone receptor complex then goes and binds to what you call in general HRE, which I talked about. What is HRE? A special part, a special sequence in the DNA where what can bind?
Hormones can bind hormone responding elements.
Hormone responsive element. This term is with reference to the DNA. An element means a part of the DNA, segment of the DNA, which has a sequence of nucleotides, which is capable of binding the receptor. But receptor by itself cannot bind that because the DNA binding site of the receptor has to be exposed. Then only it can bind. So that happens when the hormone binds to the receptor. The receptor undergoes the conformational change. It now becomes conducive of binding to the hormone responsive element. HRE is a general term saying that a segment of the DNA which is responsive to hormone. It could be any hormone. So different hormones have their own responsive elements. So those genes which are going to be controlled by, say, estrogen, they have estrogen responsive elements ahead of the gene. Some of the genes which are being controlled by corticosteroids, they will have corticosteroid responsive elements ahead of the gene. And similarly, there are hormones which can be controlled by androgens. Those genes ahead of the gene will have androgen responsive elements and so on and so forth, or progesterone responsive elements and so on and so forth. So responsive element in a segment of DNA whose nucleotide sequence can bind a hormone receptor, of course, in a hormone receptor complex. So here, receptors are present in the nucleus, and when the hormone binds to the receptor, this hormone receptor complex can then associate with the hormone responsive elements in one or more chromosomes. See, there are the number of genes on different chromosomes which can be respond to a particular hormone. So all those chromosomes will have the genes on those chromosomes. They'll have the specific hormone responsive element. At all those places, this hormone receptor complex can bind. That's the meaning of it. Understood? Understood?
Yes, yes.
Okay. Examples are what? Thyroid hormone, retinoid hormones, vitamin D. These are some of the important hormones which have a nuclear action through nuclear receptors. Now, here is generally the classical concept of the endocrine gland, which is a hormone, and this hormone then binds to a receptor. This is the target cell. The large rectangle here is the target cell. On the surface, you have a binding with the receptor. Now, what happens is binding of hormone with the receptor triggers one of the following intracellular events. When a protein hormone binds to the ligand binding domain of the N-terminus in the extracellular domain of the receptor, it brings about, and again, it's a protein. Actually, anything when bind to a protein, it disturbance conformational change. Here, the transmembrane domain The ligand this undergoes a sudden change. The receptor undergoes a conformation change at its three terminals. That triggers off a series of actions. Like, for example, the intracellular event that can be mediated by the binding of a hormone to its receptor on the surface is it can alter channel permeability by acting on pre-existing channel called those channels so for the channel in the closed state, it's movement or in or out or efflux again. What's happening? Oh my God. Okay, continue.
Sir, your voice is
Second
breaking.
is act through. What happened? My voice is breaking? Oh my God. Now it's okay? Hello?
Yes, now it's okay.
Yes, sir.
Okay. The second is will or acts through second messenger. The first effect of a protein hormone is may change the permeability of an associated ion channel. The second is it can generate a second messenger. Right? Classically, most of the protein hormones generate so-called second messengers. Right? Now, can you tell me what is a second messenger and what are the different types of second messenger that you know of?
Cyclic AMP.
Cyclic AMP?
nucleotides? Yes.
Cyclic nucleotide means?
Both CGTP and
CAMP and CGMP.
Yeah.
Cyclic AMP, cyclic GMP. Anything else?
Calcium.
Calcium. Okay, then.
IP3.
IP3. Okay. And then something related to IP3. DAG. So all these could be secondary messengers, second messengers. Okay. So some of the protein hormone on binding to the receptor will generate second messengers in second messengers. Now, the second messengers in turn will alter the structure and function of pre-existing protein. In the generally, there are again enzymes, enzymic proteins which are inactive enzymes, so-called kinase. They are essentially kinases. And by the way, I forgot to ask you what is a kinase.
Sir, the molecules enzyme that help in phosphorylation.
For that?
The enzymes that help the molecule to phosphorylate means which creates phosphorylation.
Okay. So kinases are phosphorylating enzyme. That you are saying, no?
Yes, sir.
So why not I call it simply a phosphorylase instead of calling kinase? The function of a kinase is essentially to phosphorylate a target molecule. So why should I call it kinase instead of simply calling a phosphorylase? That's a very simple term to understand. What is the phosphorylase for that man? There are phosphorylases, right? If you have studied carbon metabolism, you take glycogen phosphorylase. No? Yes or no?
Yes, sir.
So what is that phosphorylase? What is it doing?
So
and
maybe
continuing
the
on.
source.
Sir.
No, sir. First it activated and then.
I couldn't get it. What is it? What is it? Tell whatever you said, whoever it is.
So kinases add phosphorus from any energy source, whereas phosphorylase directly adds inorganic phosphates.
Okay. A phosphorylase is an enzyme which will phosphorylate a substrate by adding an inorganic phosphate, right? Whereas a kinase is again an enzyme which phosphorylates a substrate using high energy phosphate taken from ATP. So all kinases are associated with ATP, cofactor ATP. Now, for example, can you give a classic example of a kinase which you studied in your biochemistry classes?
Sir, protein kinases.
अरे बाबा मैं biochemistry में okay, let us say glucose metabolism. Glucose is metabolized.
Yes.
Glucokinase, right?
Hexokinase.
What is the factor required along with that? When you write the equation, you add something along with that. ATP added to that plus ATP is required.
Yes.
A glucokinase cannot function without ATP. Because glucokinase is an enzyme which phosphorylates glucose by taking high energy phosphate from ATP. So ATP is a necessary cofactor, whereas ATP is not a necessary cofactor of phosphorylase. So both kinases and phosphorylases are essentially phosphorylating enzyme, but fundamental difference is the ordinary phosphorylation is ordinary phosphorylase is an enzyme which adds simply an inorganic phosphate, no energy. The substrate does not get activated by that phosphorylation, right? Whereas the substrate gets highly activated when high energy phosphate is attached to the glucose molecule or any substrate, right? So the kinases are there. Now, in this case, when you talk about signal transduction through protein hormones, the enzymes are essentially, when there are different types of kinases based on the broad category
Hello.
Ma'am, we are not able to hear.
I think, yeah, just a minute. Sir's connection has a problem. Sir, can you hear us? Hello? I think he's rejoining. Let's wait for a few minutes. Yes, sir. Sir, you will have to unmute yourself.
Okay?
Yes, sir.
Okay.
Yes, he can hear, but you will have to present the slide again.
Oh my God. This is too much. I'm tired. I have to present the slide again.
Yes, sir.
Share screen. This one. Are there? No?
Yes, sir.
Yes,
Okay.
sir. Yes,
Okay.
Yes, sir.
Okay. Second is acting through second messengers, which will alter the pre-existing protein, or activation of specific genes to cause formation of new protein. I told you some of the protein hormones may also have a nuclear action. So some of the active protein kinases which are formed here will translocate into the nucleus and phosphorylate some of the transcription factors and bring about transcription of some genes. Okay. So some protein hormones may also have a nuclear action, but of course the hormone does not go into the nucleus. It is acting in the cytoplasm, secondary messenger generated, that will activate the protein kinases, and some of the phosphorylate proteins can migrate into the, some of the protein kinases can migrate into the nucleus, phosphorylate transcription factors, whereby they activate the transcription factors and bring about the transcription of some of the genes. That means there is gene expression there, and so some new protein is in the site. And ultimately, the sum total of all this, whatever happens, either in the cytoplasm or nucleus, is some physiological response that is manifested in the target cell. Then, now how lipid-soluble hormones work. Binding to specific cell receptor in the cytoplasm and form a hormone receptor complex, which will diffuse into the nucleus. And then after entering into the nucleus, the receptor may be eventually released for reuse, and the steroid activates a specific gene to produce mRNA. That's the genic action. The mRNA pass out into the cytoplasm and initiate protein synthesis. The whole process is called mobile receptor hypothesis. We already talked about, in which a steroid hormone is not attached to the plasma membrane, but is seen to move freely in the cytoplasm to the nucleus. Right, that's what it is. So essentially, through the receptor, the hormone receptor complex will bind to hormone receptor responsive elements and bring about gene expression. So most often, most of the steroid hormones go into the nucleus and activate some of the genes. That's why we say, by and large, steroid hormones have a genic action or intranuclear action. Then step one, when you look at the action of lipophilic hormones, steroid hormone, free lipophilic hormones that means hormones not bound with the receptor in the plasma membrane, diffuses through the plasma membrane into the target cell, and inside the cell, it binds with the receptor, which is intracellular and located inside cytoplasm or in the nucleus. The step 2 is after binding, each receptor has specific binding region with the hormone. That's a hormone binding domain. And then another region on the receptor, which is for binding with the DNA. So a steroid hormone receptor has 2 binding domains. One is for binding of the hormone, and the other is to bind with the DNA. Now, the DNA binding domain becomes active only when the hormone binds to the receptor. Okay. So receptor alone cannot bind to DNA unless it binds to the hormone. Once the hormone is bound to the receptor, the hormone receptor complex binds to specific regions of DNA known as hormone responsive elements. This is a general term. It can now specifically refer to them as estrogen responsive elements. If it is estrogen, progesterone responsive element is progesterone, corticosteroid responsive element is a corticon, is a hormone which is bound to the receptor, and so on and so forth. Then third is after binding to the DNA, the hormone receptor complex will activate the transcription process because it has an influence on the promoter region. You know what we call as the basal transcription factors are bound to promoter region. So the basal promoters, the basal transcription factors are activated because of this, and so the gene gets switched on. So transcription of the gene occurs, and as a result of transcription, mRNA is produced, which is processed, of course, into mature RNA, and it is passed out of the nucleus into the cytoplasm through the nuclear pores. And then once in the cytoplasm, the mRNA gets translated by the ribosome, and then the protein or enzyme is synthesized, right? And this protein that is now produced, it could be enzymatic protein or structural protein, will then mediate the functions that it's supposed to. So some change occurs in the cell as a result of the new protein that is synthesized. Now, this is a very general picture showing serum molecular action. The blood vessel, so you can see this, the protein-bound hormone, because steroid hormones cannot freely be transported through the plasma and the blood. So naturally, it must have proteins bound. So generally, the plasma protein or steroid, serum proteins are the carrier molecules for steroid hormone. So here is the protein hormone complex which is being transported in the blood. When reached at the target site, it will dissociate into free hormone molecule. All right, the carrier is separated out. So anytime you see in the blood, and you talk about steroid hormone, there will be bound hormone and free hormone, right? More the bound hormone, less is the action of the hormone. More the free hormone, more is the action. So at the time of hormone action, when it reaches the target site, it dissociates from the bound protein, and it is a free protein, naked hormone, which will then mediate the action. So as you can see here, the hormone is produced by dissociation from the bound protein. It traverses through the plasma membrane, enters into the cell. This is the entire yellow part of the cytoplasm, and this is the nucleus. So this is, of course, this part is showing the cell surface receptor of also. Sometimes, as in the case of progesterone controlling oocyte maturation, it's got a receptor or surface receptor even. So the surface receptors usually mediate. Now, for example, if you compare the protein hormone receptors which act through surface receptors and steroid hormone receptors which act intracellularly at the nucleus, you look at the action. The protein hormone actions are quick, very immediate, because they're generally controlling functions within the cytoplasm, increasing metabolic activity, stopping metabolic activity. Some physiological functions have been altered. It's very quick. Basically, the protein hormone actions are very quick, but then short-lived. Whereas the nuclear hormone action, that is steroid hormone action, will take time to bring about the effect. Once it is there, is a long-lasting. So steroid hormone responses take a little longer time to express, but the protein hormone responses are quick and immediate. It's something like the comparison the neural transmission and endocrine control of function. The neural functions are quick, immediate, short-lived, but endocrine modulations are longer in time and persist for a longer time. So here it is. So here is the hormone, which then binds to the cytoplasmic receptor, forms the hormone receptor complex, and then it translocates into the nucleus. This hormone receptor complex then binds to a DNA, what we call a hormone responsive element, brings about the transcription of the adjacent gene, produces an mRNA, which gets translated out, transported out to the nucleus and cytoplasm, and on the ribosome, they're translated, the protein is produced. This protein will bring about a physiological change or functional change in the cell. Okay, then once the function is over, the receptor and the hormone can also be dissociated. Sometimes in some of the steroid hormones will directly enter the nucleus through cytoplasm, pass through cytoplasm, and there it combines with the nuclear receptor, again binds to hormone responsive elements. So whether a cytoplasmic receptor or a nuclear receptor, steroid hormones ultimately bind to the responsive elements in the form of hormone receptor complex. Okay, it says most hydrophobic steroids are bound to plasma protein carriers. Only unbound hormone can diffuse into the target cell. Steroid hormone receptors are in the cytoplasm of either it may switch on some gene or it may suppress the expression of certain genes. It can complete. Then activated genes create new mRNA that move back into the cytoplasm. Translation produces new proteins to control new cell processes, and some steroid hormones also bind to membrane receptors that use second messenger system to create a rapid cell response, as in the case of the oocyte maturation controlled by progesterone. So this is a very simple picture. Now, how do water-soluble hormones work? A water-soluble hormone is the first messenger. As the first messenger, the hormone is the first messenger, which binds to the receptor. It passes through the blood, through the interstitial fluid, and then binds to the receptor, the exterior surface of a target cell's plasma membrane. The hormone receptor complex activates a membrane protein called a G protein. That means here, essentially, we are referring to a G protein coupled receptor. The activated G protein, in turn, activates adenylyl cyclase. Now, I don't know whether I have time to go into the G protein structure. You know, the G protein coupled G alpha, G beta, gamma complex is there bound to GT by GDP. It's the G alpha which dissociates from alpha gamma, beta gamma. The beta gamma has separate function. The G alpha is the one which translocates across the membrane and then activates adenylyl cycle, associated membrane, adenylyl cycle of the membrane. The G gamma activates. Even the G gamma, the different types of G gamma, stimulatory, inhibitory. Some of the G alphas will stimulate the function. Some of the G alpha will inhibit the function, so forth. I don't think I can go into those things today. Should I finish this? All the time. What do you think? Hello?
Finish it.
Okay.
Sir,
Okay.
you can take it 5-10 minutes extra. That's what I was telling.
Oh, really? Thank you. Water-soluble hormone, the first messenger, diffuses from the blood through interstitial fluid and then binds to the receptor at the exterior surface of a target cell's plasma membrane. The hormone receptor complex activates a membrane protein called G protein. The activated G protein, in turn, activates. What you need to understand is once the G protein on the cytoplasmic domain of the receptor is activated, it in turn activates the adjoining enzyme. See, adenylyl cyclase is a membrane-bound enzyme on the inner side of the membrane. So this G protein moves, makes contact with adenylyl cyclase and activates the enzyme adenylyl cyclase. Now, adenylyl cyclase, what is the function of adenylyl? Converting ATP to cyclic AMP. So adenylyl cyclase converts ATP into cyclic AMP because the enzyme's active site is on the inner surface of the plasma membrane, I told you. This reaction occurs in the cytosol of the cell. Now, cyclic AMP, the second messenger that is so generated, activates one or more protein kinases, which may be free in the cytosol or bound to the plasma membrane. That even these protein kinases may be lying either free in the cytoplasm or some of them may be even attached to the plasma depending upon the functions, okay, functional requirements. In different target cells, you have different versions of this. Now, ultimately, the question is when you say the cyclic AMP activates one or more protein kinases, how would cyclic AMP activate a protein kinase? That's the question. See, when you look at the protein kinases, it has got two parts, a regulatory subunit and a catalytic subunit. You understand the difference between the two?
Yes, sir.
The protein kinases have a regulatory subunit and a catalytic subunit. It is through the catalytic subunit it can activate protein kinases. Is it clear? Or the protein kinase can function in phosphorylation. Now, a cyclic AMP, what it does is when the catalytic subunit is bound with the regulatory subunit, it is inhibited. It is inactive. The catalytic site of the protein kinase remains inactive as long as it is bound with the regulatory subunits. So the regulatory subunit, that means the regulatory subunits have two binding sites. One is the binding site for the catalytic sites of the protein kinase. The other is a binding site for cyclic AMP. When cyclic AMP is generated in the cell, it will go and bind with binding site on the regulatory subunit of the protein kinase. And this binding, the moment the cyclic AMP binds the regulatory subunit, conformation changes. It can no longer bind to the catalytic site. It becomes free. The cyclic AMP regulatory subunit dissociates from that. The catalytic subunits become free. The moment the catalytic subunits become free, they become active. They start what? They become active protein kinases, will start phosphorylating target proteins in the cell. So cyclic AMP activates one or more protein kinases, which may be free in the cytosol or bound to the plasma membrane. A protein kinase is an enzyme that phosphorylates or adds a phosphate group to cellular proteins such as enzyme. The donor of the phosphate group is ATP, which is converted to ADP in the process. Okay. Then now activated protein kinases. You know what is the way in which a cyclic AMP activates the protein kinase? The protein kinase remains inactive in the cell bound with the regulatory subunit. ATP, the cyclic AMP goes and binds the regulatory subunit. It dissociates. The catalytic subunit becomes free. It becomes active. It starts phosphorylating target protein. Now, once the protein kinase phosphorylates one or more cellular proteins, now phosphorylation activates some of these proteins and inactivates them, rather like turning a switch on or off. That means some of those target proteins which are being phosphorylated by a protein kinase will either become active on phosphorylation or will become inactive on phosphorylation. Is it clear? Do you understand? Now, when you talk about glucagon action liver cells, what is glucagon? Yes. Hello?
A hormone secreted by pancreas.
Right. Where does it act?
In liver.
In the liver cell.
In the liver. So obviously it is the surface receptor, it's the protein hormone. So it binds to the receptor. Then what happens? What happens when a glucagon binds to the receptor on that target liver cells? What is ultimately activated?
Glycogenesis.
That is the function. Glycogenolysis is the function that is active. But how can glycogenolysis occur? What is glucagon actually doing? Anything that happens in the cell requires an enzyme. Even a single reaction, addition of hydrogen or removal, requires an enzyme. Without enzyme, no reaction can occur in a cell. So when you talk about glycogenesis, the process of breaking down glycogen, it cannot happen without the mediation or participation of an enzyme. So what is the enzyme that is required for breaking down glycogen? You know, who was the person who discovered the action of glucagon on liver cells? No? See, both glucagon and insulin act through hormones, through enzymes. Ultimately, they are going to mediate the function of two enzymes. What is the enzyme that is being activated by insulin, and what is the enzyme that will be activated by glucagon? So you're not studied the classical work on protein hormone action, and that's essentially glucagon. Who was the scientist who worked on glucagon action and bringing about glycogenolysis in the liver? Any idea? No?
No, sir.
Oh, that's too bad. Sir. That's too bad. The very classical things you must remember. How can you not study those things? You studied biochemistry? You studied the process of glycogenolysis? Huh?
Yes, sir.
So
yes,
when you study glycogenolysis, tell me whatever you studied about glycogenolysis. How does it happen? You simply said glycogen is the process by glycogen broken into glucose. That's it. You studied. That's it. Right? That's what you studied. That the 12th standard student also will tell me glycogenolysis is the breakdown of glycogen into glucose. That's it. But how does it happen? Is enzyme required? Is any enzyme required for that? What is that? Yes?
Epinephrine.
Epinephrine also can. That is, I'm asking about the glucagon action first. It's and epinephrine action will be almost similar. Epinephrine also can mediate glycogenolysis. But that's not the question. The question is, what is the modus operandi of either glucagon or epinephrine mediating glycogenolysis? That's my question. How does it bring about?
Phosphorylation of glycogen.
Phosphorylation. Huh?
Sir, protein kinases, maybe.
Glycogen phosphorylase.
Glycogen phosphorylase. Yes, protein kinase. Before that, the protein kinase is the one which is phosphorylated. Ultimately, the enzyme glycogen phosphorylase is phosphorylated or dephosphorylated? Phosphorylated?
Yes.
So it becomes an active phosphorylase now?
Yes.
And that will mediate glycogen breakdown?
Yes, sir.
And what about insulin? What does it do?
It dephosphorylates this enzyme.
Dephosphorylation of?
This enzyme,
which enzyme?
Glycogen phosphorylase.
That means the glycogen phosphorylase. Oh, that means glycogen phosphorylase, when it's phosphorylated, it will mediate glycogenolysis, and when it is dephosphorylated, it produces glycogenesis. That's what I'm trying to say?
No, sir.
No. ये तो अभी ऐसे बोला आपने. I didn't say anything. That's what you said. Now I'm asking you, what is the mode of action of insulin? Similar to phosphorylase, which is activated by glucagon, is there any enzyme which is activated by insulin to bring about glycogenesis? Nah.
In phosphatase.
Huh?
Actually, sir, insulin inactivates adenylyl cyclase. So by that, there is no CMP production, which in turn
Adenylyl cyclase
inactivates
activated, no
protein
CMP
kinase
production. क्या बोल क्या रहे हो? Ultimately, what happens? How would my question is very simple?
Glucose to glycogen.
Yeah, that I know. That is called glycogenesis. How does it happen? That's what I'm asking you. And that's what the insulin ultimately activates. Again, enzyme is required to join together glucose molecules to form a glycogen. It requires an enzyme.
Hexokinase.
Sir, glucose hexokinase, not glucose hexokinase, hexokinase.
Fantastic. So instead of synthesizing, you're going to break down of glucose. वाह, बहुत अच्छा है. तुमको साउथ में जाओ बोलेगा तुम नॉर्थ में जाएगी, बहुत अच्छी बात है ना? When glucose molecules that we join together to form glycogen, you're talking about glucokinase and fructokinase or a hexokinase. They are participating in glucose breakdown.
I'm not sure, but it is glucose synthase or something.
Glycogen synthase. Glycogen synthase.
Yes.
Glycogen synthase, an enzyme which will synthesize glycogen. It is that enzyme which will be activated by insulin. Now, it's an homework for you. You please go back and check up what is the pathway through which glucagon acts and ultimately activates phosphorylase, and what is the pathway through which insulin acts and stimulates glycogen synthase. That you have to find out. Okay?
Okay, sir.
Okay. Now, what's the question? I cannot complete. Now, what do you do? If anybody is interested, you can give me another time, sometime later, we can do all those things. What do you think?
You can take.
When? I can take when? You decide and then give me a time. Of course, it cannot be done when this the whole course is going on. After this crash course, you can give me some time. Whenever you want, you let me know a time, then I can give you. I can do this.
Sir, I think
Please share your mail ID too.
Okay.
Sir, I think I will just write an email to everyone, and
those who are interested can respond. Let me know.
Yes,
on the date.
We will fix up a date after, or maybe not this Sunday, but the next
after
Sunday.
finishing this workshop only.
Okay, sir. Fine.
But in between, I
So-
don't want to get into it.
Okay, sir. There was one more topic which was remaining from your last session.
That also,
So we
gene
can
regulation
plug-
also.
Yes,
So
sir.
those who are interested for both the topic, you can inform me at time. I can complete for you.
Sure, sir. So I'll communicate to everyone.
Yeah, it
and
is on their
accordingly we'll
interest.
fix
If you are interested, you are
Yes.
free. I am ready to help you.
Okay, sir.
If you are not
Sure, sir.
interested, then I'm interested. Not interested. What is it?
Sir, we are interested.
You are
Sir,
interested.
we are
Very good. Show your
interested.
interest. Then give me the time. I'll take it for you.
Yes, sir.
Yes,
We
sir.
will
Okay.
revert
Yeah.
back
Okay,
to
sir.
the mail.
Okay.
Yeah.
Okay. Fine.
So
Thank you very
I'll
much.
fix up
Okay.
a time and send an email to all of you.
Yeah. Okay. Sure.
Okay.
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 Fine.
Thank you, sir. So
Thank
I
you very
think
much. Thank you for
we
listening
can
to
end
me.
here.
Okay.
Thank you, sir.
Okay. Welcome.
Okay. For tomorrow,
Thank you, sir.
we'll again.
Thank you, ma'am.
Yeah. Thank you. And we'll have 2 sessions again. So see you at 10 o'clock sharp tomorrow morning. And we'll
Ma'am,
send you. Yeah.
On the Zoom itself or on YouTube?
We are keeping both the platforms open now onwards.
Okay,
So
ma'am.
it will be Zoom as well as YouTube, because we
Okay,
understand there
Please
can be
share this
connectivity
PPT too.
issues.
Ma'am,
Yeah.
yesterday's summary chart we haven't got that IUCN, no? If you could share
So
that.
we are sending that to you in some time from now. Okay.
Okay, yeah, and the
Okay.
questions
Ma'am,
also.
Yes.
the practice exercise we have got only of session 1, I
Yes,
guess.
session 2 we are sending now.
Okay, and ma'am,
Okay.
today's also you will send
Yeah, today's
today?
also we will send it.
So we
Right?
will have 3 exercises for today.
Yes.
Okay, thank
Okay.
you, ma'am.
Okay, so as and when you get time, you keep on completing and submitting it to us. Okay.
Sure, ma'am.
Yes, ma'am.
Thank
Ma'am,
you.
Okay.
please attach
Ma'am,
sir's
ma'am,
mail
ये
ID or
PPT मिल
contact
सकता
number
है?
also.
Yeah, sure. I'll
Along with
send
PPT.
Ramcharan sir's ID. And if there's anything specific you want to convey to sir, you can convey to me also, and we'll convey it to him. That's also possible.
Okay, ma'am.
Okay, ma'am.
Thank you.
Thank you.
Okay. Yeah. Okay, sir. We'll end here.
Okay.
Thank you,
Okay.
sir.
Bye. Thank you. Thank you.